Imagine peering into the genetic code of a man who walked the earth nearly 4,500 years ago. This isn't a scene from a science fiction movie. It's a groundbreaking achievement by a team of scientists who sequenced the genome of an ancient Egyptian. But here's where it gets controversial. This discovery doesn't just shed light on one individual's genetic makeup. It challenges our understanding of Egypt's early dynastic history and the migrations that shaped the ancient world. Let's dive into the fascinating journey of how this was accomplished and what it means for our understanding of human history. A team of dedicated researchers has managed to sequence the genome of a man buried in Egypt around 4,500 years ago. This study, published in a prestigious journal, offers an unprecedented glimpse into the genetic ancestry of early Egyptians. The findings reveal intriguing links to both ancient North Africa and Mesopotamia, a region that encompasses modern-day Iraq and parts of Syria, Turkey, and Iran. But this wasn't an easy feat. Egypt's harsh climate and terrain have long posed significant challenges for such genetic studies. However, lead researcher Adeline Morez-Jacobs and her team made a remarkable breakthrough. We had the opportunity to speak with her about the hurdles of sequencing ancient remains, the scientific advancements that made this discovery possible, and how this genome could reshape our understanding of Egypt's early dynastic history. But first, let's break down the basics. What is genome sequencing, and how does it work? Genome sequencing is the process of reading an organism's entire genetic code. In humans, this code consists of about 3 billion chemical letters, A, C, T, and G. The technology to sequence genomes was first developed in the late 1970s, and by 2003, scientists had completed the first full human genome. However, applying this technology to ancient remains is a different story. DNA degrades over time due to heat, humidity, and chemical reactions. Ancient bones and teeth are often filled with DNA from soil microbes rather than the individual we want to study. Early attempts in the 1980s to sequence DNA from mummified remains were largely unsuccessful because the available methods weren't suited to the tiny, fragmented molecules left after centuries or millennia. To sequence DNA, scientists need to make multiple copies of it so there's enough to analyze. Originally, this involved inserting DNA into bacteria and waiting for colonies to grow, a process that took days and yielded inconsistent results. Two major breakthroughs changed the game. In the early 1990s, PCR, polymerase chain reaction, allowed scientists to make millions of DNA copies in just a few hours. By the mid-2000s, new sequencing machines could read thousands of DNA fragments simultaneously. These advancements not only sped up the process, but also made it more reliable, enabling the sequencing of even highly degraded DNA. Since then, researchers have reconstructed the genomes of extinct human relatives like Neanderthals and over 10,000 ancient individuals who lived in the past 45,000 years. But the work remains challenging. Success rates are low for very old remains, and tropical climates destroy DNA quickly. So what makes the sequencing of these particular remains so exceptional? Our study stands out because we were able to sequence a surprisingly well-preserved genome from a region where ancient DNA rarely survives. When we analyzed the sample, we found that about 4% to 5% of all DNA fragments came from the individual himself. The rest came from bacteria and other organisms that colonized the remains after burial. In living organisms, the quantity of DNA of interest is usually between 40% and 90%. That 4% to 5% might seem tiny, but in this part of the world, it's a relatively high proportion and enough to recover meaningful genetic information. We believe the individual's unusual burial may have contributed to this preservation. He was placed inside a ceramic vessel within a rock-cut tomb, which could have shielded him from heat, moisture, and other damaging elements for thousands of years. To maximize this rare preservation, we filtered out the very shortest DNA fragments, which are too damaged to be useful. The sequencing machines could then focus on higher quality pieces. Thanks to advanced facilities at the Francis Crick Institute, we were able to read the DNA over and over, generating about 8 billion sequences in total. This gave us enough data to reconstruct the genome of what we call the Nuweirat individual, making him the oldest genome from Egypt to date. Does this open new frontiers in genetic research? We didn't develop entirely new techniques for this study, but we combined some of the most effective methods currently available into a single, optimized pipeline. This is what paleogeneticists often do. We adapt and refine existing methods to push the limits of what can be recovered from fragile remains. That's why this result matters. It shows that with the right combination of methods, we can sometimes retrieve genomes even from places where DNA usually doesn't survive well, like Egypt. Egypt is a treasure trove for archaeology, with remains that could answer major questions about human history, migration, and cultural change. Our success suggests that other ancient Egyptian remains might still hold genetic secrets, opening the door to discoveries we couldn't have imagined just a decade ago. And this is the part most people miss. The most exciting result was uncovering this man's genetic ancestry. By comparing his DNA to ancient genomes from Africa, Western Asia, and Europe, we found that about 80% of his ancestry was shared with earlier North African populations, suggesting shared roots within the local population. The remaining 20% was more similar to groups from the Eastern Fertile Crescent, particularly Neolithic Mesopotamia, present-day Iraq. This might sound expected, but until now, we had no direct genetic data from an old kingdom, 2686 to 2125 BCE Egyptian individual. The results support earlier studies of skeletal features from this period, which suggested close links to pre-dynastic populations, but the genome gives a far more precise and conclusive picture. This genetic profile fits with archaeological evidence of long-standing connections between Egypt and the Eastern Fertile Crescent, dating back at least 10,000 years with the spread of farming, domesticated animals, and new crops into Egypt. Both regions also developed some of the world's first writing systems, hieroglyphs in Egypt and cuneiform in Mesopotamia. Our finding adds genetic evidence to the picture, suggesting that along with goods and ideas, people themselves were moving between these regions. Of course, one person can't represent the full diversity of ancient Egyptian society, which was likely complex and cosmopolitan. But this successful sequencing opens the door for future studies, building a richer and more nuanced picture of the people who lived there over thousands of years. Adeline Morez-Jacobs, the lead researcher, does not work for, consult, own shares in, or receive funding from any company or organization that would benefit from this article. Her affiliations are purely academic, ensuring an unbiased perspective. So what do you think? Does this discovery challenge your understanding of ancient migrations and genetic ancestry? Share your thoughts in the comments below. Let's spark a conversation about how this groundbreaking research could reshape our understanding of human history.